Good morning, and first of all, I'd like to say thank you to the organizing committee for giving me this opportunity. So this talk is about antigen presentation in acute GVHD. So this is a graphic summary of today's talk, and then please let me show first to you. As you know, the naive T cell are the mediate GVHD. They are going to the lymph node or target organ directly, and then they are presented antigen by the recipient, mostly recipient APCs. And then it's included hematopoietic APC or also the non-hematopoietic APC, which are not really appreciated so far. But today's half of my talk is about the role of the host non-hematopoietic APC in GVHD induction. Then the T cell is presented around proliferate and then differentiate to the Th1 or Th17 and then start the damage of the organs. So in parallel, the donor APC is deconstituted the hematopoietic APC. So once started the tissue damage, in especially colon, the donor DC is stimulated to proliferate and then activate via the damage or pathogen-associated molecular pattern, which is the innate signals getting to the damaged tissue. And then those donor DC migrate into the mesenteric lymph node. And in this process, the subset of the donor DC is really important. And then they are present antigen to T cell again here. And then T cell is proliferated and then activated and implant the integrin alpha 4 beta 7, and then which is the ligand of the MADCAM1 expressed on the inflamed mucosal tissue. So they back to the intestine and then makes the severe GVHD. So this is the GVHD enhancement process, and then my second half talk is about the role of donor APC in this enhancement. So donor T cell presented the host-derived antigen, alloantigen, and then activated by the peptide MHC complex. However, the MHC class 1 and the class 2 are differently expressed in the recipient. As you know, the MHC class 1 is expressed most of the nuclear cells. However, the MHC class 2 is quite limited, like hematopoietic professional APCs. So thus, experimental models have been developed so far as the MHC class 1 dependent, means the CD8 T cell mediated GVHD, or MHC class 2 dependent CD4 T cell mediated GVHD. So the question is, which cell type, donor or host, or hematopoietic versus non-hematopoietic cells present our antigen to the donor T cell to induce or enhance GVHD? So in concept, there is 3 types of the APCs. So after the conditioning regimen, recipient hematopoietic APCs start reduced. And then, of course, after the transplant, they are more reduced and then replaced with the donor hematopoietic APCs. However, there is a non-hematopoietic APC exists, and then they are always recipient type. So for the CD8 mediated GVHD, host hematopoietic APC are necessary and sufficient for the GVHD induction. And then donor APC are required for the maximal GVHD. For CD4 mediated GVHD, MHC class 2 expressed on the host hematopoietic APC is sufficient to elicit CD4 mediated GVHD. And regarding host hematopoietic APC, the recipient DC are sufficient to induce CD4 or CD8 mediated GVHD. So likely the recipient hematopoietic APC is really important. However, for the CD4-mediated GVHD, it's not really proven that this cell type is really necessary. This, in other words, the host hematopoietic APC are when host hematopoietic APC are unable to present antigen, is GVHD reduced? So this is the important question for the perspective of the therapeutic point of view. What are the important APC driving the MHC cluster dependent GVHD? To ask this question, we used the BLAC6 by DBA2 model with the TCR transgenic CD4 T cells. This transgenic T cell recognized the antigen from the DBA2 recipient on the host or recipient BLAC6 type of MHC cluster. So when the donor is a wild-type BLAC6 mouse, they can present with the appropriate MHC cluster to this transgenic T cells. And also wild-type BLAC6 by DBA2 mice also can present. However, when the donor have MHC cluster deficiency, they cannot present. And also when the recipient is a BLAC6 MHC cluster deficient by DBA2 mouse, they have antigen derived from DBA2. However, they can't present because there is no appropriate MHC cluster. So with this mice as a different combination, we can compare to the host and the donor antigen presentation, or the host antigen presentation only, donor antigen presentation only, or nothing antigen presentation to these transgenic T cells. This showing the T cell expansion as a bioluminescent signal. And as you can see, the T cell expansion is quite equivalent between the host and the donor antigen presenting group versus and the host antigen presentation only group. However, with the donor antigen group, antigen presentation only, they can't expand well. And this, however, is a quite similar direction. The host and the donor antigen presentation group and the host antigen presentation only group have a severe GVHD. However, the donor antigen presentation only could not induce this GVHD. When the T cell dose really escalated, the donor APC antigen presentation can induce a partial GVHD, but much less extent. So the antigen presentation by host APC is really required. Then we want to know which cell type of the host APC is important. Quantitatively, the host DC is important, so that's why we compare to the donor DC depletion, host DC depletion versus non-DC depletion using the CD11C-driven diphtheria toxin receptor expressing recipient with the diphtheria toxin induction administration. Surprisingly, the host DC depletion increased the effector T cell expansion compared to the non-depletion. And this, however, is no difference, finally no difference. So this means the depletion of host DC does not prevent a response to the alloantigen. So other cell types must drive the donor T cell activation. So this raising the question if the host hematopoietic APC are really required for the GVHD induction. Then we ask the question compared to the hematopoietic antigen presentation versus non-hematopoietic antigen presentation. Here is the forearms, but all mouse doesn't have the antigen presentation by donor. And the difference between forearms is a host hematopoietic or non-hematopoietic antigen presentation. When the host non-hematopoietic and the hematopoietic antigen presentation are intact, the survival, GVHD survival is like that. And then also non-hematopoietic APC only also have a severe GVHD. Surprisingly, the host hematopoietic APC only can induce the much less GVHD. And then again, here is the T cell expansion, and the host hematopoietic and the non-hematopoietic APC group have a good expansion of T cells, and then non-hematopoietic APC group is similar. However, the hematopoietic APC only group has much less expansion of the T cells. So host non-hematopoietic APC are sufficient to induce lethal GVHD. Actually, indeed, the MHC cluster is expressed on the non-hematopoietic cells. This is a small intestinal villi, and then showing the MHC cluster as a green, and the CD45 hematopoietic marker as a red. Hematopoietic MHC cluster expressing cells is shown as a red yellowish color like that. But here is clearly the green single positive cells, means a non-hematopoietic MHC cluster expressing APCs. So in conclusion, the first part is host non-hematopoietic APC is crucial to induce the CD4 T cell mediated GVHD. So now moving on the second part, after the GVHD is already induced. What we know about the donor APC, so donor APC are not required and are inefficient for the GVHD induction as shown in the previous slides, but they are required for the maximal disease. In the donor conventional DC are the most potent APC capable of presenting our antigens. So we know the donor APC exacerbates GVHD, but we don't know the further mechanism, who, how, and where. So to ask this question, we developed a model to assess the Antigen presentation by donor APC exclusively. So these irradiated biopsies for transplanted bone marrow and T cell as a GVHD group, or the T cell depleted bone marrow as a non-GVHD group from the allogeneic black 6 mouse. And then on day 12, when the APC is fully reconstituted by the donor APC, they report a T cell which has the luciferase signal injected, and then a couple of days later, we can look at which organ have this T cell expansion. This allows us to compare the antigen presentation by donor is how different between the GVHD versus non-GVHD circumstance. And what we know found is in GVHD mouse, especially, they have a mesenteric lymph node, have a T cell starting a T cell expansion, and then a few days later, this T cell expansion is expand the whole intestine and also other systemic organs. It means the donor antigen presentation during the GVHD is initiated in the mesenteric lymph node. Then we look at which cell types is important in the donor APC, and the donor DC is preferentially presented antigen in the mesenteric lymph node, whereas the donor macrophage are dispensable for the antigen presentation. Damage associated or pathogen associated molecular pattern dump or pump signalings enhance antigen presentation in the mesenteric lymph node during the GVHD. So next question is, how do the DC and the innate signals link to each other in the mesenteric lymph node? So we look at the donor DC in the mesenteric lymph node. The DC gated as a major class CD11C double positive operation, and then we can separate 3 subsets. Here is a CD103 single positive or CD11B single positive or double positive population. What we found is a CD103 positive, 11B negative population showing as red here have the most highest capacity to present our antigen. Sorry. Yes. Highest capacity of antigen presentation, and also the highest capacity of IL-12 production. Also, they have a highest CCL7 expression means that this molecule is required for the migration from intestine to the lymph node. So then we look at, compared to the wild-type bone marrow, the recipient of wild-type bone marrow versus recipient of the mediated TRIF, double knockout bone marrow or raised knockout bone marrow. These molecules are down pump signaling transducers, and the mediated TRIF are the signal transducer for the toll-like receptor or inflammasomes, and the RAGE is the receptor for the nuclear protein HMGB1 or heat shock protein or nucleic acid. So these molecule deficient mouse have much less percent absolute number of CD103 positive 11B negative fraction. And then this results in the reduction of antigen presenting cell and then reduction of cytokine producing cells, DC in the mesenteric lymph node. So the mesenteric lymph node CD103 positive 11B negative DC present alloantigen and they secrete IL-12 under the influence of innate signals. So next question is how does the down pump signaling influence the CD103 positive 11B negative population? Because the mesenteric lymph node is a draining lymph node of intestine. And then we look at the donor DC distribution after transplant. This signal is come from CD11C driven donor luciferase. So that's why, not same to the previous slide, this showing the donor DC expansion. And then we compare the GVHD versus non-GVHD. As you can see, that after day 12, not much different, but after that, the GVHD mouse have accumulate the mesenteric lymph node DC and also in colon. It's not happening in non-GVHD mouse. So that's why we look at the donor DC in the colon. And as you can see, the donor DC in colon, in particular the CD103 positive, CD11 negative fraction, have again antigen presentation and the IL-12 producing population here. It's not exist in the non-GVHD mouse. So GVHD induced and the increase and the activated donor DC in the colon. So final question is, do CD103 positive 11B negative DC present alloantigen in the colon or mesenteric lymph node following their migration, because this population exists in colon and mesenteric lymph node. Thus, we compared the recipient of wild-type bone marrow versus CCL7 deficient bone marrow. As you can see, the absence of CCL7, donor DC are normal in the colon, but fail to migrate to the mesenteric lymph node here, missing population. And then this results in the T cell expansion in the mesenteric lymph node, and then intestine didn't show here. And also, it's important for the implant alpha-4 beta-7. So wild-type group have a high expression of alpha-4 beta-7 integrally on the T cell, but not happening in the CCL7 deficient bone marrow transplanted mouse. So colon-derived donor DC migrate to the mesenteric lymph node via the CCL7 and then implant the alpha-4 beta-7 integrally on the donor T cells. So second part of the talk is the conclusion is after the GVHD induced, tissue damage allowed to enter the pathogen or damage-associated molecule, and then they stimulate the donor T cell expansion, and then activation in the colon, especially the CD103 positive 11B negative population, which is equivalent to the human CD141 high DC, but this population is migrating the lymph node and then stimulate the T cell expansion, and those T cells implant back to the intestine, and then this cycle is the feed-forward cascade to make the severe GVHD. So in conclusion, the host non-hematopoietic epithelial cells are crucial for the induction of CD4 T cell mediated GVHD. The mechanism of antigen presentation utilized by the host non-hematopoietic APC can be targeted for the prevention of GVHD. For that, we need to identify the cell types of the host non-hematopoietic APC, and also we need to elucidate the intracellular pathways to be functional APC. Also, the enhancement process, the chronic donor-derived DC determine the severity of GVHD. Since GVHD target organs do not have the leukemia and the donor DC do not mediate severe effect, these strategies are targeted the host non-hematopoietic APC or donor chronic DC should also spare the GVL. Biomarker-based identification of the patient at the high risk for the severe GVHD may allow the preemptive treatment that they interrupt the chronic mesenteric lymph node T cell cycle. The candidate would be the CCL7, IL-12, IL-6, or alpha-4 beta-7 molecules. So I'd like to say thank you for all my colleagues and then collaborators, especially my supervisor and mentor, Jeff Hill, and also funding source of NHMRC Leukemia Foundation of Australia, the Koyama seed funding. Thank you very much for your attention.
Thank you, Jamie, and I wish to thank the scientific board of the meeting to invite me and to allow me to discuss with you the role of the microbiome in GVHD. Now, the idea of manipulating the microbiome in the setting of allogeneic stem cell transplantation is not a new one. We have this old data that mice grown in a germ-free condition do not develop graft-versus-host-disease after transplantation, at least of marrow. You see 100% survival if mice are grown under germ-free conditions. If the mice are brought back to a conventional surrounding germ-free only for 8 days, almost all mice die from GVHD. And if it's intermediate at day 26, you can see 1/3 of mice die from GVHD. These were data from Van Beckum. He later confirmed the role of the intestinal microflora, which we now call the intestinal microbiome, also in rhesus monkeys, and there were other data in dogs. And finally, the first data, a small data series in patients, that this germ-free conditions could help to prevent GVHD, and since that time decontamination is still one approach we are using in many centers currently. On the other hand, we know that antibiotic prophylaxis in acute leukemia and stem cell transplant patients has an impact on all-cause mortality and a reduction of febrile episodes. So the rationale was prevention of GVHD and infections for prophylaxis. Now we have to be aware that these data and these findings were based on analysis of bacteria which were culturable. And it's now clear that maximum of 30% of the microbiota in our body are culturable. So with the introduction of new techniques to characterize the microbiome in a culture-independent way, like the high-throughput sequencing of the 16S ribosomal RNA, we could get a completely different view on the microbiota of the whole body, but especially of the gut. This was part of the Human Microbiome Project. In summary, these projects described a large proportion of commensal, mainly anaerobic bacteria like Clostridia species, Bacteroidetes, and they showed that these were associated with protection from inflammatory and autoimmune disease. We now know that 30 trillion human cells encounter 100 trillion microbial cells in the body. And what is most important, the current estimate is that we have 1.5 million metabolites from these microbial cells which interact with the human cells and govern many of the physiological processes in our body. Now, when we now applied these new culture-independent techniques like the 16S rRNA analysis to stem cell transplant patients, we saw a completely different picture. We saw in both studies an early loss of intestinal microbiota diversity. The first study came from the Sloan Kettering from Robert Yank. They showed both in mice and humans that if they developed graft-versus-host disease, then there was a major loss within the Clostridialis and Lactobacillus group. And they also showed if they pre-treated mice with broad-spectrum antibiotics, this induced a microbiome shift, and this reduced in accelerated GVHD with increased pathology and reduced survival. We also performed a series of microbiota analysis in 31 patients, and again we observed the early loss of the commensal bacteria, especially from the Clostridialis group. And in our setting, we saw a strong abundance, in some patients even 100% abundance of enterococci, which was caused by the prophylactic and therapeutic antibodies, and was even worse if in the presence of gastrointestinal GVHD. Now, these changes have prognostic significance. Here, the New York group divided the patients according to the diversity indices found in the microbiota samples 15 days after transplantation. And you have a group with high diversity, which means they have a lot of commensals, and a group with low diversity, which means there are a few genus left, and commensals are mainly gone. And you can see you have a big difference in survival after 3 years, which is mainly to non-relapse related mortality. This was later on specifically shown for the genus Plautia by Robert Yank. Again, abundance of Plautia would associate with high diversity and protect against GVH-related mortality. Now, our group used a different approach to analyze microbiota diversity, and we went back to the metabolites produced by this bacteria. And one interesting group of metabolites are indoles. Indoles are derived from tryptophan in the gastrointestinal tract. If commensal bacteria have tryptophanase, they can convert tryptophan to indole. This indole is taken up by the gut, is transported to the liver, and in the liver you have sulfatation, so you have an indoxyl sulfate as a product, and this indoxyl sulfate is then excreted to the urine, where we can measure a bacterial metabolite. We came to the indoxyl sulfate because there were studies comparing germ-free mice and mice in conventional surroundings, and one of the completely absent metabolites in germ-free mice was this indoxyl sulfate. So we can now say high indoxyl sulfate levels in the urine indicates many commensals, high diversity, and low indoxyl sulfate, few commensals, low diversity. Now, when we did a serial analysis of indoxyl sulfate levels in 130 patients at different time points after transplantation, we observed that low indoxyl sulfate levels, again very early in the first 10 days after transplantation, predicted the long-term outcome. You can see here the patients with low indoxyl sulfate levels. Remember, this means low commensals, low diversity, and the treatment-related mortality of patients with high indoxyl sulfate levels means high diversity, and again it's predictive. Early loss of diversity is associated with poorer outcome. When we asked for the factors affecting this indoxyl sulfate levels, we saw that prophylactic and therapeutic antibiotics influenced the indoxyl sulfate levels, and we also found a well-known genetic risk factor, the NOT2 card15 SNPs, which we had reported 10 years before, which represent a receptor associated with antibacterial defense. So here is a link to our previous work. So let's go in detail to prophylactic and later on to therapeutic antibodies. Now, as we observed this almost complete shift to enterococcal flora, and we even had an outbreak of vancomycin-resistant enterococci at the time, in our 16S analysis, we decided to stop our previous decontamination with ciprofloxacin metronidazole and switched to rifaximin, a non-absorbable antibiotic. As you can see here, the mean early indoxyl sulfate levels were much higher in the rifaximin group. So again, this leaves obviously some of the commensal Clostridia. And as you also can see here, in the rifaximin group, the mortality was lower, and this translated also in a quite different outcome 3 years after transplantation, with a GVH, acute and chronic GVHD-related mortality of 40%, almost 40% in the old decontamination group, and of 19% in the rifaximin group. Now let's go a step further to antibiotic treatment. Here we did a combined analysis together with the colleagues from the Sloan Kettering Center, and we analyzed 621 patients undergoing allogeneic transplantation, T-repleted transplantation, and we analyzed the impact of timing of antibiotic treatment on outcome. We made 3 groups: an early antibiotic group, which received antibiotics before the day of transplant, late antibiotic groups, which received antibiotics on the day of transplant or thereafter, and we had a no antibiotic group with no systemic antibiotics given throughout the transplant. The striking result was that there was a highly different treatment-related mortality with regard to the timing of antibiotics. The lowest treatment-related mortality, and look, this is 80 months after transplantation, was in the no antibiotic groups, those patients who did not need systemic broad-spectrum antibiotics, and the highest mortality, up to 50%, in the early antibiotic group. Many colleagues said, okay, the early antibiotic groups are patients with far advanced diseases. They have a lot of additional risk factors. So we did a variety of risk analysis, risk scores, Karnofsky index, performance index, the stage at the time of transplantation. But this effect of early use of systemic broad-spectrum antibiotics showed up in each risk group, even in the low-risk group, indicating that's an independent risk factor for outcome. How does this work? Again, we looked at the microbiota, and we see that we had a different composition of the commensal microbiota. Here you see the indoxyl sulfate levels in the early antibiotic group, in the late antibiotic group, and the no antibiotic group, again at day 0 to 10. And I have shown you that this is a risk factor for poor outcome. And both the New York group and also our group also made direct analysis on the commensal bacteria. Here is one example. We have a PCR for Clostridial group 14A, and we applied this to the samples of our patients. And you can see the late no antibiotic group is close to 10 to the 9 copy numbers, which is close to the status of normal patients. But the early antibiotic group has lost this Clostridia, again lost the commensal group of bacteria. Similar observations were very recently published by a Canadian group and by a pediatric group, showing that depletion of Clostridia is associated with increased mortality, and they have also shown this in a murine experiment. Now, when we ask for the mechanisms, Jamie mentioned this already, we come to metabolites produced by this bacteria. We know I mentioned the huge interaction of bacteria with human cells by protective metabolites, and there's also huge interactions between the different bacteria and the species in the gut. So we have one group, the indoles, you know them already, produced from dietary tryptophan, and they are known to support survival of mixed microbial communities. They are known to induce colonization resistance. This is a part of the mechanism of colonization resistance we frequently discuss in infectious disease models. But they also have impact on the epithelial cell barrier properties. They strengthen it. They are anti-inflammatory at the cytokine at T cell level, and a very important aspect is indoles bind to the aryl hydrocarbon receptor of innate lymphoid cells type 3 and then induce the production of interleukin 22, which is an important cytokine for protection of epithelial cells. Now, even more, we recently started experiments. We're looking for the immunomodulation by indoles. Sakila Gimiri from our group incubated dendritic cells with LPS in the presence or absence of indoxyl sulfate levels as we find it in the urine and in the serum of patients. And to our surprise, this indoxyl sulfate suppressed the pro-inflammatory phenotype of dendritic cells. IL-12 goes down at the cytokine level, at the RNA level, and on the other hand, IL-10 goes up, and we saw similar effects for CD80 and CD86. And when we used this indoxyl sulfate incubated dendritic cells in mixed lymphocyte cultures, there was much less interferon gamma production, and even in some cultures a rise of regulatory T cells. So this is a clear example how commensal bacteria try to tolerize the human immune system in order to live in a peaceful coexistence. The other large group of protective metabolites are the short-chain fatty acids, especially butyrate is here important. Again, they are important for epithelial integrity, like tight junction and survival of epithelial cells. And again, they are anti-inflammatory. They downregulate IL-12, they downregulate co-stimulatory molecules, and they can induce regulatory T cells. This is shown by the group von Ann Arbor and New York. They have used butyrate gavage to protect against GVHD, and they also introduced a 17-strain cocktail of Clostridia in the GVH model. As you can see here, the 17-strain cocktail induced higher butyrate levels in the lumen and in the cell wall of the intestinal wall, and if added in a GVH model, it protected against GVHD to some extent as compared to the GVH model without this 17-strain Clostridia. So again, we have here Clostridia producing protective metabolites and protecting against inflammation. Now, when it comes to GVHD, Jamie mentioned this, then there is Paneth cell damage in addition to the disturbed microbiota. And we asked the question, drives this further loss of microbiota diversity? Here you can see the number of Paneth cells in biopsies from patients without GVHD and with severe GVHD, and you see the strong decrease of Paneth cells, and we have now tested the antimicrobial peptides in the biopsies, alpha-defensin 5 and 6, and also REC3 alpha. Here are patients without GVHD and patients with GVHD, and you see that both antimicrobial peptides are going down, which is in accordance with the loss of Paneth cells. And the consequence, the functional consequence, is seen here when we analyzed the indoxyl sulfate levels at the time of biopsy. Those who had no GVHD had higher indoxyl sulfate levels than those who had GVHD, indicating that the microbiome is even further disturbed at the time of graft-versus-host disease. So, taking all this together, we can think of GVHD as a disturbed balance of microbiota, but also disturbed balance of cells regulated by microbiota, like innate lymphoid cells, like regulatory T cells. And of course, the question is, can we modulate this? And there's a huge variety of options, and it's summarized here. I talked about antibiotics, so we can change the decontamination strategy. We even can skip decontamination. We should think about our timing of antibiotics, or whether we use commensal sparing antibiotics. There are other strategies blocking bacteria, pathogenic bacteria. We recently published on this oral immunoglobulin. Then we could go to prebiotics. So commensal bacteria need non-digestible carbohydrates like fibers, and there are several trials under the way using fibers as additional nutrients in the context of stem cell transplantation. We could use postbiotics. I mentioned the beneficial effect of indole and short-chain fatty acids, and this would have the advantage that they are not just destroyed by antibiotics. And of course, we could change the food. And we can use probiotics, directly apply microbiota, like in the fecal microbiota transplantation, and on the long run, defined consortia will be developed. That this really works was recently shown by two small publications where the colleagues from Japan and from Graz used fecal microbiota transplantation from healthy related stem cell donors in 4 patients with steroid resistance or steroid-dependent GVHD. Here they had a healthy unrelated donor. In both studies, transfer of fecal microbiota was safe without any severe adverse effects, no infections. And it was striking that in this series there were 3 complete responses and 1 partial response in steroid-refractory GVHD with restoration of commensals, and also responses in the Graz series. Now, finally, I would like to mention that microbiota play not only a role with regard to graft-versus-host disease and toxicity of stem cell transplantation. There is more and more evidence that also the efficacy of cytotoxic treatment and also of checkpoint inhibitors is strongly influenced by microbiota. In this study by Nathalie Pflug, they showed that lymphoma treatment with DHAP gives 20% less survival if you concomitantly block gram-positive bacteria with vancomycin. I think we will have to consider a lot of things in our hematology oncology setting in the future. A very interesting recent paper, again from the New York group. There is also association of specific microbiota with relapse. They found that abundance of the Eubacterium limosum, another commensal bacterium, associates with reduced risk of relapse, and it will be very interesting to see whether these are specific T-cell antigen interactions which contribute to GVL effect, or what is the mechanism behind this protection from GVL, even by the microbiota. I hope I could give you an overview about the current role and view on the microbiota in the setting of allogeneic transplantation. And I finally want to thank my research group, especially Daniela Weber and Sakila Gimire, our collaborating groups, both in New York, Marcel van den Brink's group and Jamie Ferrara's group. In the local level, Peter Oefner, who is running the urinary ES assays, and Andre Gessner, who is doing the 16S sequencing. And of course, I would like to acknowledge the grant support by the DFG, the Jose Carreras Leukemia Foundation, and the European Commission. And I wish to thank you for your attention.
Okay, so I'd like to finish by sharing with you both clinical and experimental data regarding, first, the role of biomarkers in predicting GVHD outcomes, and then what some of those biomarkers are teaching us about the biology. So what you see here are data from the bone marrow transplant clinical trials network, where we had 300 patients who had provided samples at the onset of GVHD. And the graph shows you the non-relapse mortality based on the presenting grade of GVHD for those 300 patients. And essentially those grades are not significantly different from each other, and this reproduces data from many studies that show that the clinical severity of diagnosis does not correlate with NRM and often does not guide treatment. When we looked at the biomarkers in those blood samples, we found that 3 of them, ST2, Reg3A, and the TNF receptor type 1, could be combined into a biomarker signature with an algorithm that would weight each of the concentrations of the biomarkers and produce a predicted probability for each patient of non-relapse mortality. We could then rank order the probabilities and divide the patients into 3 separate groups, which we called the Ann Arbor staging system, since we had done that work in Ann Arbor. Grades 1, 2, and 3. You now see again these same 300 patients according to their Ann Arbor stage. I'm not showing you the relapse mortality, but the probability did not predict relapse, and as a consequence, the overall survival from these 3 groups was significantly different. And these data were published 2 years ago by, with John Levine as the first author, in Lancet Hematology. We used the non-relapse mortality as the endpoint for defining the algorithm, but as you would expect, the patients who had the low-risk group, who were in the low-risk group, would have the greatest response to treatment, particularly to steroid treatment. So here you see the complete response rate at day 28 in the 3 risk groups, about 75% for Ann Arbor 1, about 50% for Ann Arbor 2, and slightly less than 30% for Ann Arbor 3. What was most interesting in these biomarkers was the fact that they would, in the same group of 300 patients that were in the validation set, if a patient presented with Glucksberg 1 or modified Glucksberg 1 that's less than 50% of a rash, body surface area rash, what you see is 24% of the patients stayed at low risk, more than half of the patients became intermediate risk, and about 20% of the patients became high risk. And here you can see the non-relapse mortality for each of those patient groups. This same proportion of patients appeared also in the Glucksberg 2, so this is mild diarrhea as well as a more severe skin rash or liver disease. What you see is that we have approximately the same proportion of patients. Slightly more than half are in the intermediate group, a quarter are in the low-risk group, and about 20% in the high-risk group. And perhaps most surprisingly, even those patients who presented with more than a liter of diarrhea and who would be considered overall grade 3 by this grading scale, a quarter of them were actually low risk, about half of them were intermediate risk, and a quarter remained high risk. So just if you would remember this going forward, according to this 3-biomarker signature, 3-biomarker algorithm, slightly more than half of the patients are still in the intermediate risk group, about a quarter are in the low risk group, and about 20% are in the high risk group. So this is the slide that you've seen before. We've heard about how the host antigen presenting cells are very important. We've got these donor T cells that are activating. The single best biomarker in all of the studies that have been performed, both by our groups and others, the single best biomarker is ST2. The vast majority of ST2 comes from the GI tract, and it comes from stromal cells and endothelium, some epithelium as well. So we believe that this is being released very early. ST2 is itself a sort of decoy receptor. It's a soluble receptor for IL-33. So what you see is that the cell becomes activated. IL-33 is an alarmin. So most of it is going to, when it actually gets into the bloodstream, is going to be bound to the soluble receptor. You've heard from Ernst Holler how the commensals are and their metabolites are very important and early on. We're now going to focus in this next little bit on one of the other biomarkers, which is reg3, which is produced by Paneth cells, as you heard from Ernst. It is stored in the mucus. It's a heavy molecule, it's stored in the mucus, and then and it is driven in part by interleukin 22, that is secreted by these ILC3 cells. So reg3 and ST2 are going to wind up being major players in the next part of the talk. We knew from the algorithm at the onset of GVHD that many of the cases, for example, in that low-risk group, we could predict GI GVHD before there were symptoms. Those biomarkers were already in the bloodstream before we actually had GI symptoms. So we asked whether we could develop a new algorithm using the same 3 biomarkers, and in addition, we're going to put in the IL-2 receptor alpha, which is another single excellent biomarker. It just fell out of the algorithm for the onset staging. So we're going to see whether we can predict non-relapse mortality and steroid-resistant GVHD prior to the onset of symptoms. So we're going to try to develop an algorithm at day +7 after transplant to predict severe GVHD and non-relapse mortality. For this, we're going to use samples from MAGIC, the Mount Sinai Acute GVHD International Consortium. The participating centers are shown here: Mount Sinai, the University of Michigan, Mayo Clinic, Ohio State, University of Pennsylvania, Emory, Regensburg, Hamburg, Dresden, Würzburg, and Bangkok. So this is a multi-continent, multi-center data set, which we deliberately have made so that we could try to develop an algorithm that would be applicable in any patient in any hospital. The data that I'm about to show you have now been published 2 weeks ago in JCI Insight. First author is Matthew Hartwell. So we use competing risk regression to model 6-month non-relapse mortality with relapse as the competing risk. The computer models compared all 13 possible combinations of algorithms of 4 biomarkers, of all the biomarkers, of all 4, for the most accurate prediction of 6-month NRM. We then used Monte Carlo cross-validation to test 75 different training sets that had been randomly generated to find the best combination of biomarkers. So there was one combination with all 4. The best combination of all 3 biomarkers was ST2, REG3 alpha, and IL-2 receptor alpha. The best combination of 2 biomarkers was ST2 and REG3. The single best biomarker was ST2. The best algorithm, which identified most accurately the largest number of high-risk patients, was the 2 biomarker combination of ST2 and REG3 alpha. So the final algorithm generates an estimated probability of non-relapse mortality for an individual patient, and then we needed to define thresholds that would divide the high-risk from the low-risk group. There were several such thresholds, and we chose the final one that gave us the largest number of high-risk patients with the maximum difference in non-relapse mortality between the high-risk and the low-risk groups. So remember, this is a single day 7 after transplant blood test. No patients have graft-versus-host disease yet. Here's the training set on the left and the test set on the right, about 600 patients and 309. You see the overall non-relapse mortality is 11 and 12% respectively. The algorithm gives us a difference of about 20% in terms of the overall the difference between the high-risk and the low-risk groups, and it's about 16% of the patients. That's essentially the best it's going to be, because that's what the computer modeling is doing over and over again with logistic regression. So the first test is our test set. Here you see that there's actually a 23% difference, and it's approximately 17% of the patients. The validation set was those were all patients combined from the University of Michigan and Regensburg. The validation set is now from 9 centers in those 3 continents. It's an N of 358. There's an overall non-relapse mortality of 13%. We now have a 16% difference between high risk and low risk, and now it's about 20% of the patients who are identified as high risk. I'm not showing you the relapse data. They are published in the article, but there was no prediction of relapse, and therefore the overall survival is highly significantly different between these 2 groups. If we look at individual pre-transplant clinical risk factors, such as related donor or unrelated donor, what we see is that the algorithm works equally well. So in the related donor group on the left, you see that the algorithm gives you again a 20% difference between mortality, and 12% of the patients are identified as high risk. Whereas in the unrelated donor group, you have again approximately 20% differences in NRM, but now we've identified 21% of patients as high risk. So because we believe that the biomarkers are actually showing us subclinical GVHD damage, we're already incorporating into the algorithm some of these preclinical risk factors, and therefore there's high fidelity of the algorithm within each risk group. And this, we found similar results as are published, whether we looked at donor type, the age of the patient, the use of antithymocyte globulin as GVHD prophylaxis or the intensity of the conditioning regimen. As you would expect, these algorithms do indeed predict specific GVHD outcomes. So this is acute GVHD-related mortality, which we define either as death from GVHD or death from infection while being treated for GVHD. So that's approximately 4-fold higher in the high-risk group. And here is steroid refractory GVHD. The difference is also approximately 3-fold higher in the high-risk group. Those were day 7 samples. We now asked whether that same algorithm, looking at 2 biomarkers, could give us the same risk stratification of patients at the time of onset of GVHD. So now we're going to essentially go back and do the Ann Arbor grades for these patients, but we have to look at samples that are taken at the onset of GVHD symptoms or at the onset of treatment. So in that was a large data set that I just showed you. It was about 1,300 patients. In 212 of those patients, we had onset samples. So we took them, we measured the biomarkers, and we used now the 2 biomarker algorithm and used the thresholds that would give us the same mortality strata in the Ann Arbor group, and this is what we found. We can indeed separate using now 2 biomarkers. We can separate patients at the onset of GVHD into high, intermediate, and low risk for mortality. These are highly statistically different. In fact, the 2 biomarker algorithm is an improvement over the initial 3 biomarker algorithm because now almost 50% of the patients have been defined as low risk Ann Arbor 1, and about 27 or 28% are in the intermediate risk and the high risk. This is also published, but as you remember from our from the first slides, it was Ann Arbor 2 was the most with about 55%, and Ann Arbor 1 was then only 25%. We believe that the difference, the improvement here is largely due to the fact that between the first study in 2015 and this study, the commercially available kit that measures ST2 is now significantly more sensitive. We are now measuring picograms instead of nanograms, and that has given us a greater distinction, particularly in the patients between the low-risk group and the intermediate risk group. This algorithm also works after treatment. So Sunday, you will see a full presentation of these data, but we looked at about 400 patients 1 week after their treatment for GVHD from within this group. We're now using the same 2 biomarker algorithm. Here is our test cohort of 236. There's a 27% mortality. The algorithm divides the low-risk group into 12% NRM versus 50% for the high-risk group. We essentially see the same separation in our test, in our validation cohort. In data that I'm not showing you, we get very similar separations, whether patients have had a response to treatment on day 7. If they've had a response, it still turns out about half of those patients are at high risk and are going to have a high NRM. And even if they don't have a complete response by day 7, you can still see a significant number of those patients are actually at low risk. So now we have a single algorithm that can tell us about the future of GVHD outcomes before the onset of symptoms, at the onset of symptoms, and after treatment. So in the last couple minutes, let's talk a little bit about the biology. This is now early acute GVHD as opposed to very early. We see that these cells are damaging the intestinal stem cells that are known to be targets. Here's a Paneth cell that's divided. The ILC3 cells that produce the interleukin-22 are also decimated. There's a little bit of a paradox here. These are unpublished data now. If a patient has GVHD, we see that there are fewer, these are clinical data, there are fewer Paneth cells, there's less GVHD actually by staining, but there's a higher level in these same patients, there's a higher level of reg3 in the bloodstream. We went to a mouse model to understand this. Black is GVHD, white is not. And what you see is that in the animals without GVHD, there is this massive upregulation of reg3 throughout the villus, whereas if the animals do have graft-versus-host disease, there's actually very little. That suggested that reg3 was important. So if you now do a transplant in a reg3 knockout animal versus a wild type, everything is the same, same donor, same recipient, except that the recipients can't make reg3. You see that there's a dramatic decrease in survival. You have even fewer Paneth cells if you use that as a semi-quantitative marker for histology. But the Paneth cells themselves are okay because they can reconstitute if there's no GVHD after the transplant. So it's not a Paneth cell defect per se. Here is H&E staining versus by the way, it's reg3 gamma in the mouse, it's reg3 alpha in the human. Those are the homologs. Here's a donor. You can see normal GVHD histology, no GVHD, normal histology, a little bit of reg3 staining. Here you can see the villus blunting of GVHD, and now that there's much less reg3 by immunohistochemistry. And if you give IL-22, which is the cytokine that is released by those ILC3 cells, you can see that there's restoration of the villus length, and now the entire villus length, essentially all of the epithelium is expressing reg3. And when you express reg3 in the epithelium and you essentially reestablish the barrier, the enterocyte barrier, in fact, so this is GVH, this is phosphate buffered saline. If you give IL-22, you're increasing reg3 in the GI tract and you're decreasing it in the serum. So the reg3 that we see leaking into the serum is essentially a surrogate marker for crypt damage. We then went on to say, does IL-22 require reg3 to work? So here is a wild-type animal with GVHD and getting phosphate buffered saline. You can see just a little bit of reg3. If you give IL-22, you restore the villus length, and you see lots of reg3. Here's the knockout animal. The GVHD is even worse. There's no, as you would expect, there's no reg3 expression when you give IL-22. Here, these are the interferon gamma positive T cells, which are a marker of effector cells in the gut. You reduce them in the wild-type animals, but you don't change them in the softer colors, both for CD4 positive cells and CD8 positive cells. Both of them go down in the wild-type, but not in the knockout. And as a result, you can rescue these mice. In the wild-type, this has been shown before by Alan Hanash and Marcel Vandenbrink. You can rescue mice by giving them IL-22, but not if you can't make reg3. And that's true both in a minor antigen mismatch model of GVHD and a fully allo model. So reg3 is required for the beneficial effect of IL-22. So this opens up now, not only do we have biomarkers to look at early stages of GVHD, it starts to focus our attention on the innate immune system that's present in the GI epithelium and how we might be able to use it in terms of trying to restore health to these cells. So you can imagine now that IL-22 is inducing Reg3, it's preserving what Paneth cells, enterocytes, and intestinal stem cells you already have. And this now takes us away from just immunosuppressive strategies to proactive reconstitution strategies of the innate immune system in the GI tract. So we've developed a 2-biomarker algorithm that predicts NRM and lethal GVHD in over 1,000 patients. It predicts lethal GVHD early at onset of symptoms and 1 week after treatment. The algorithm predicts outcomes independently of pre-transplant risk factors or GVHD grade or response to treatment. It may be able to guide preemptive treatment, perhaps specifically aimed at the GI tract, and IL-22, which comes from damaged ILC3 cells during GVHD. Restoring it can help treat GVHD, but it requires the induction of Reg3 gamma. Let me thank my collaborators, John Levine, Matt Hartwell, who did the predictive work, Hannah Major-Monfried, who's talking on Sunday about the response after treatment, Ernst Holler, Umut Uzbek, our statistician, Dongchang Zhao, who did the animal work, and our funding agencies. Thank you very much.